Welcome to the second Emerging Biotech Company Showcase, hosted virtually by NewYorkBIO and the New York Stock Exchange. The showcase brings together the financial community and a group of New York-based emerging biotech companies that are developing novel therapeutics and technologies that could revolutionize patient care. As part of the event, we're posting conversations with the leadership of these companies that gives more background and color behind the management team and their mission. Special thanks to our sponsors at Fox Rothschild, JP Morgan, Carta, Newmark Knight Frank, VWR, and Solberry Trout. Welcome to a deeper dive with David Baram of Emendo Biotherapeutics. So, David Baram, it's fantastic to have you with us and to have Emendo be part of NewYorkBIO and the New York Stock Exchange's second annual Emerging Biotech Company Showcase. And I'm really glad you could join us for a deeper dive. And what we like to do on these is I like to get a little bit more color around the starting of the company. So Emendo is something where you've put together a platform with really a more precise editing tool for CRISPR-based therapies. So was the motivation to start the company more on the technological side, or can you walk us through kind of where the motivation for starting the company came from and what the aha moment was?
Sure. First of all, thank you, Derek. Thank you for the opportunity to speak here. And yeah, I'll be glad to answer that. So Emendo was actually established mid-2015. At that time, CRISPR was, it's kind of the very beginning. CRISPR was first published for human applications somewhere at mid-2013, so that was 2 years after. And we came at that point and we were looking at this very exciting technology. As mainly protein engineers, that's our scientific backgrounds comes mainly from the areas of synthetic biology and protein engineering. So looking at that, we try to identify the challenges of this space for going forward and making it, making therapeutics out of it. And the first thing that we put our finger on, and it's obvious, is of course the potential off-target effect, its risks. I'm sure that if you look at the gene therapy space, and I'm sure you're familiar with that, there was a kind of a decade of a delay. It just came because of safety issues that were, due to off-target effect, right? With the skid, with unfortunate results of the treatment for SCID. So it was clear to us that in order to make this a therapeutic, safety would be first, and to get high precision is not enough. You want to get the highest possible precision in order to make a therapy out of it. It's actually, I see it as an obligation. It's not even something, you have to do that. Eventually you inject it to humans. It has to be super safe. So at that time, one thing that we have identified, besides the need for, let's even call it extreme precision, is another understanding that we had back then, that we don't think a one solution fits all approach would be enough here. What does that mean? At the early days, even for me, the first time I thought of CRISPR as a potential application, I thought, wow, this is amazing. You have this part with nucleic acid, the guide. It takes you, it's like a cassette that targets any location along the genome. And the rest of it is pretty easy, as opposed to other alternatives. For instance, talons were there and meganucleases were there, and they were all protein, right? So in order to direct them to a certain place, you had to do a lot of work, and this is easy. It's like a cassette. Yep. But then, like biology always shows you, it's never that easy. It never actually works like that. So we realized that precision is a big issue. Activity is also a very significant issue. And when I'm saying we didn't believe that one solution fits all, we were looking at engineering as a solution, but we realized early on that rather than providing, engineering the nucleus to have to generally be more precise on multiple targets, it made much more sense to get to extreme selectivity or extreme precision if you engineer the nucleus to be as precise as possible against a specific target. And
Right.
that's where, yes, please.
So basically, you're engineering the nuclease for each specific target that you would want to hit. Ergo, you don't take the general broad limitations of CRISPR nucleases. You look at therapeutic indications and say, for this, we need this specific type of nuclease. The nuclease has to do X, Y, and Z for this to be valuable for this set of patients.
Exactly. That is actually exactly the case. Now, there's a challenge associated with that, and it's cost and timelines, right? Because now, as opposed to the previous perception where you just pick a guide and a high fidelity, highly active CRISPR nuclease, mix them together, you have a composition, and go ahead. This is an approach that says no. If you want to get the highest possible precision and the highest possible activity, you need to optimize every composition, right? So the challenge here is making it cost effective and fast enough. And that's exactly what we did from the very beginning. We have established a platform that is based on both computational approaches, computational algorithms that we have developed in-house, and an experimental part that is based on high throughput methodologies in the lab. We combine both of them in order to be able to optimize this composition in a very short time. By very short time, I mean that it will take us between 3 to 6 months to fully optimize a composition per a specific target to have extreme fidelity and activity. Now, it doesn't end with that. We got that, and by the way, we have quite a few examples and data cases where we show that with this approach, we were able to get rid of some stubborn off targets. Some off target sites are just stubborn. It's very hard to get rid of them. None of the high fidelity solutions that are there, that exist off the shelf, were able to deal with that. Applying our approach, we were able to eliminate these off targets. Eventually, this allows us, as we see it, to target any site along the genome. We will first choose a cut site, then we will make the composition be safe and effective at this specific site. But first, we choose the best cut site, or the most, we'll choose the cut site according to the editing strategy and not according to fidelity and activity constraints.
Yeah. Can you talk a little bit about building out the early team? So I imagine you incubated some of this at the Weizmann Institute, right? And with your background, you're certainly no stranger to early stage companies. So can you talk about how you got the initial team together and what your team looks like now? Because now you have sites both in Israel and New York.
Right. So as you probably read, but for anyone who listens to it doesn't know, my scientific background is structural biology and protein engineering from the Weizmann Institute of Science. Actually, I did my thesis under the guidance of Ada Yonat. She's a Nobel laureate for 2009. So this was fun parts. Emendo was actually established by a group of graduates of the Weizmann Institute. By chance, they're all from my class at the Weizmann Institute of Science. Eventually, I decided to get into this space had the initial idea, and I gathered some of my most talented classmates from different disciplines, by the way, and I gathered them from sometimes from different parts of the globe in order to establish the first scientific team that actually founded Emendo. So we gathered protein engineering, structural biology, computational biology, DNA damage repair expertise for the founding team. It was also fun because we were friends as well. It wasn't that hard for us to get the initial funding, and just we funded it ourselves at the very beginning, and then Series A, and we started building the additional team. Today, Emendo is a much much larger company compared to what we were before. We are more than 70 employees at the moment and growing. We have different teams and divisions in computational engineering, proof of concept approaches, and then of course drug development. It's we have a great team here, in Israel and in the US. BD, financials, and a lot of the development is actually done in the US, where research and the things that are more let's say early stage are done in our research facility next to the Weizmann Institute of Science.
Mm-hmm. Well, that's terrific. So you were able to strike what I would call an early informative partnership with Takeda. Do you envision that kind of partnership to be one of the ways that you expect to grow the company? It's a way to potentially get into broader areas than you might be able to with just internal capital.
Yeah, definitely. First of all, with the early collaboration or partnership with Takeda was extremely valuable for Emendo, because at the early stages, when you have to set your directions, you want to choose the right indications. Being able to have this discussion with big pharma early on, I'm talking about very early, Series A, that's great. That's amazing, because eventually you have to approve a drug here. With such a new technology, you have to take so many factors into account. And since the company was established by scientists, you initially, you love the science. So you challenge yourself with the science. So partnering early on with a pharma company that is product oriented, not science oriented. Of course, they have great science, but they are product oriented. I think that was great to have that early on, and they were part of our discussion, and they gave us a great directions early on. Going forward, it's important to say that we have no exclusive relationship with Takeda whatsoever, and we would for sure. And we are actively working on establishing additional strategic partnerships within the industry. And actually, we are, I'll say with all modesty, we're a great company for that, because you have Emendo, and we see it as a kind of a factory with an end-to-end solution. If you come with a target gene for Emendo, let's say that you're a pharma company, you want to target a specific gene, you're interested in the disease, you already have a drug that is sold in this specific space, and the gene therapy, gene editing will become a competition, you want to develop that, we are a great partner for that, because we can take a challenge of just give us the gene, and we will, just with our, two platforms that we're working on, we will be able to provide you with an optimized composition where we handled everything, the editing strategy, the off targets. We have everything in-house, all of these capabilities in-house, and of course the optimization. So therefore, if I look forward and I look at the gene editing space, I think the gene editing is I know, maybe the gene therapy of 7-8 years ago. That's where gene editing is, maybe even less. And I assume that big pharma will be more comfortable in making, significant deals in gene editing and CRISPR gene editing, probably from 2 years to 5 years from now. And I plan to make Emendo ready for that time, because eventually it will make more sense to get a company or to partner with a company that has a lot of expertise with doing gene editing, rather than starting learning from the very beginning.
I mean, one of the biggest milestones is really, being able to show clinical effects in a patient group. And I had wondered if you had started to either reach out to patient foundations or other sorts of advocacy groups to, perhaps position this type of technology a bit closer to patients from the get-go. Because anytime you think about genetic diseases, very oftentimes you're actually talking about children, and it's certainly an area where being closer to the patient and understanding their journey is really key, whether it's in clinical development or the eventual rollout of a therapy.
Yeah, of course. And since we haven't discussed our pipeline of indications, but let's talk just about the first indication that we chose to develop a cure for. And again, I love saying that. I love the word we're developing a cure. We're not developing a treatment. This is a cure, potentially one-time treatment will lead to a cure. So that's actually Elaine, or severe congenital neutropenia, orphan disease. Around 3,000 cases in the US. And the first thing that we have done is contacted Professor David Dale from University of Washington, who has been for many, many years probably the go-to center for Elaine patients. And he also takes a very active role in the patient organization. So definitely yes. And not only for this is just an example. We have recruited already patients to take part in our studies, and with the help of Dr. Dale, we were able to do that. And again, we feel that this is actually, it's a unique position. It's the first time that you can come to a patient organization or even a patient personally and saying, "Hey, no, we're developing a cure here," and that's and we get very, very good responses for that. So.
I can imagine. And, I've looked through your pipeline. I think you have what looks like a great list of collaborators. You have the NIH, you have Stanford, you have Columbia. It's really exciting to see the way that you've positioned the company. I would love to talk to you a little bit about your outfit in New York. Obviously, as NewYorkBIO, we're a little biased, and we know New York is fantastic. Yeah. But can you talk a little bit about the decision to kind of set up shop in New York City, some of the things that kind of drew you to the ecosystem, and what some of your initial observations have been?
So to begin with, I think that it was probably the first decision that I have made when I decided to establish Emendo, that it's going to be in New York. Several reasons there. The gene therapy, gene editing space has been exploding for quite a few years now. But New York was never a center, right? It's Boston and West Coast also. New York wasn't a center for that, and I actually saw it back then as an opportunity because New York is an important, first of all, investor base also for for the biotech space. So you have the investors there. You're just a couple of hours away from Boston, which is a hub for gene therapy, gene editing, and you have some very very good research institutions and hospital there. We have a very strong collaboration with Columbia University on ophthalmology. This is great. I assume that if we were trying to do the same in Boston, this brilliant research group were already preoccupied with 4 different collaborations, and they wouldn't have had the ability or the capacity to do that. So I saw New York as a great opportunity to find collaborations, scientific collaborations, and we have done that, and it has been great. Another thing, by the way, that I had in mind, and actually proves itself to be true, since again, New York is there is a biotech industry within New York, but it's not as developed or crowded as it is in Boston, for instance. I think
crowded is a good word. I'd like to use crowded.
Yeah, crowded. Yeah, crowded probably better.
Yeah, crowded is good.
Yeah, but I think that the ability to get a hold of talent, to be able to recruit talented people in New York without that massive competition. Listen, one thing that is being said within the industry, there's no loyalty in Boston, for instance, in the Boston area, to companies, because so many just employees, they get they get offers all the time, and it's very hard to keep them. In New York, you have talented people that just love to live in New York. So I think there's actually an opportunity there, and we were able to get great employees there. Having Christine as our CFO, it's great to have her. I don't know, if we were in another place, it was more crowded, maybe it would have been more difficult to get someone in with this level of experience and talent.
I can't agree with you more. I mean, we know this and we live this, but it's just really great to hear somebody else echo the best parts of New York. Well, I think this has been fantastic. I'm really excited for what you're doing, and excited to have you be part of our program on Wednesday. Thank you for joining us on a deeper dive with David Baram of Emendo Biotherapeutics. We're looking forward to the Emerging Biotech Company Showcase on December 9th. Look for more information on www.newyorkbio.org and follow us on Twitter @NewYorkBIO.